Ever since the dawn of humanity, medicine and healthcare have always been something rather routine. Believe it or not, we humans are more susceptible to diseases and infections than any other animal on Earth. As we take our costly visits to the doctors, maybe because of some cold, it becomes a reset for our bodies, but it's not permanent. You see, the fevers, the coughs, and the sniffles will continue to come back, and there's no way for us to stop them. When we take our prescribed pills, of course, we're bound to feel better. But once you stop taking them and their effects wear off, it's open season again for these diseases to come back and infect us. Another thing worth noting is that not every person at the hospital is simply taking one of their costly visits. If you take into account that according to the CDC, approximately 50% of adults reported having a chronic condition that requires continuous treatment, I'd be willing to bet that the majority of the people at the hospital are actually there for their monthly, maybe even bi-weekly visits. So what does this all mean? To me, what this means is that our medical system at the moment is inefficient. A large number of people are having to continuously drag themselves to the hospital because their condition confines them to those areas. By understanding this issue in healthcare, it only makes sense that in order to solve the problem, we need to stop giving these patients even the reason to come to the hospital. We need to dig deep into the roots of the issue. And that's where genetics comes into play. Hi, my name is Toya, and today I'll be sharing with you a few ideas of how genetics is leading to the fall of traditional medicine. And so, on a molecular level, for a virus or a disease to infect us, it needs to find a way into our cells. And normally, the way they do this is by binding to receptors on our cell membrane and going through the openings that these cell membranes or receptors open up. How traditional medicine would usually fix your illness is by having you ingest a pill and hoping that pill wipes out all the pathogens from your body. But as I said before, this is just a temporary fix. The pathogen can always try to come back and try to infect our bodies again. But what if we had a more long-term solution? What if we could remove these receptors from ever appearing on our cell membranes in their entirety? And so at Toronto University's Faculty of Biochemistry, they're starting to experiment with a technique called double-stranded RNA interference, or dsRNAi in short. This biotech technique was discovered by Andrew Fire and Craig Mello around 20 years ago and won them the 2006 Nobel Prize in Physiology and Medicine. At Toronto, this technique is meant to prevent the formation of receptors on shrimp cells that will make them susceptible to infections by a bacteria called E. coli. The diagram on the screen will give you a quick rundown of how this whole process will work. And so given its name, dsRNAi, we're going to have to get our dsRNA in the cell first. And this dsRNA sequence should be identical to the gene that codes for the memory receptor. And this will be crucial later on. But once you have your dsRNA in the cell, it's going to automatically cut into these smaller single interfering RNAs, or siRNAs, and it's going to go bind to form an RISC complex. And since this genetic material on the RISC complex is complementary to the gene that codes for the memory receptor, labeled as an mRNA on the screen right there, it's going to act as a guide for this complex to go locate that gene in the cell and effectively break it down. And so what this does is it prevents that gene from ever being translated into the memory receptor or the protein, resulting in its absence from the shrimp cell membrane. Great. Well, how is this process any better than just taking your complementary or traditional pill? Well, if you notice, since the receptors on the shrimp cell are gone, not only does it limit the cell's chance of being infected down to absolute zero, But it also acts as a more long-term solution, as these dsRNAs can stay inside the shrimp cell for an extensive period of time, providing consistent protection against E. coli. So even during this time, if E. coli manages to get inside the shrimp's body, it still can't get inside the shrimp cell, making it completely harmless. And so, staying on the topic of bacteria, one of humanity's most pestering issues right now that leads to so many frequent hospital visits is the constant evolution and adaptation of bacteria. You see, antibiotics like penicillin are meant to stop vital processes that go on inside bacteria, resulting in their death. But because of bacteria's rapid ability to evolve and develop a resistance to these antibiotics, we're actually running out of solutions to combat bacterial infections, while the bacteria themselves are just getting stronger and stronger. I can imagine one of the questions you're probably asking yourself is, well, why can't we just make new antibiotics? Well, unfortunately, since the golden age of antibiotic discovery, which was around 60 years ago, barely any new antibiotics have emerged for commercial use. And this can be attributed to how the science and chemistry behind finding a new chemical that effectively kills bacteria is incredibly difficult, time-consuming, and expensive, often taking around 15 to 20 years and over $1 billion just to find a new antibiotic. And so it seems like all hope is lost in our battle against bacteria. But we humans don't ever give up, do we? And so introducing you to what's called the Microbe Lab, run by Professors Vorapon Chaikiratisak and Professor Pushit Nonchui. I had the fabulous opportunity to interview Ajahn Vorapon a couple months ago. And what I learned from the Microbe Lab, on my part, was rather uplifting regarding the situation we're facing right now. And so, fueled by the release prediction that by the year 2030, more than 10 million people will die from these antibiotic-resistant strains of bacteria known as superbugs, Professors Vorapon and Puchit started the Microbe Lab in order to investigate new ways to combat these superbugs. And just to put into perspective for everybody, the amount of people who die from cancer yearly also hovers at around the 10 million mark. So it just goes to show how big of an issue these superbugs can become in the future. So these dedicated scientists at the Microbe Lab experiment with something called bacteriophages. They're essentially viruses that target and kill bacteria specifically by injecting their genetic material into them and taking control of the cell. They call themselves phage hunters, who essentially hunt, collect, and organize these phages into a database for future use, instead of antibiotics. But then this whole proposition and proposal raises another concern. What if our bacteria become resistant to the bacteriophages as well? I think it's crucial to note that a bacteria's resistant genome, or its information bank it can store, is finite. Think of it as a library. If a bacteria wants to store a new book and develop a new resistance about how to fight against a certain bacteriophage, then it may have to get rid of another book that was previously stored there that tells it how to fight against an antibiotic. If it wants to keep new books, then it has to get rid of some old ones. And so to ensure that our bacteria are being killed every single time, despite its pre-existing immunities, scientists can inject multiple phages, and maybe even an antibiotic as well, into the bacteria, in hopes that it lacks the recipes to just one of these attacks. And such a concoction is what's called a phage cocktail. And this is exactly how I feel the Microbe Lab will continue to excel in the future. By providing doctors and healthcare professionals with the chemical compositions and details to a wide variety of bacteriophages, it makes their jobs a lot easier when designed to treat patients with bacterial infections and save lives that could have been lost to these superbugs. And finally, I want to finish off with another simple example of how genetics is changing the world of healthcare, but this time in present day. Have any of you in the audience ever heard of the disease called sickle cell anemia? If not, you may have heard of its Southeast Asian cousin, thalassemia. Thalassemia is a genetic disease that causes the blood to not make up enough of a protein called hemoglobin. It's what allows the red blood cells to transfer oxygen around the body. As a result, red blood cells lacking hemoglobin have this shriveled appearance. What a thalassemia patient needs to do right now is they need to get blood transfusions every 3 to 4 weeks to ensure that their blood is prepared to transport oxygen around the body. And as you can imagine, this is incredibly time-consuming and inconvenient. And it also digs a financial hole for many patients. Before I tell you what scientists are currently working on to alleviate the symptoms of thalassemia patients, I want you to understand that red blood cells are unique cells and that they can't divide. What this means is that if I were to put the correct hemoglobin gene into the source of all these red blood cells, then eventually all the faulty red blood cells would die out over time. And you'd be left with only the fully functional red blood cells that are constantly being produced over and over again. And this is exactly what you call gene therapy. And so researchers at a company called Bluebird Bio have discovered that if you take a viral vector or virus that contains the correct hemoglobin genes and have it infect a thalassemia patient's bone marrow, or the source of all the stem cells that produce red blood cells, then eventually all the red blood cells that circulate inside a thalassemia patient will be fully functional. Although this technique is currently priced at a whopping 40 million baht for us Thais, doctors and researchers at the Faculty of Medicine, Ramathibodi Hospital, Sri Raj Hospital, and the Institute of Molecular Biosciences, Mahidol University, are currently working on reducing this price down to just 10 million baht. And still, this sounds like a huge sum of money. But if you take into account that blood transfusions every 3 to 4 weeks for 70 to 80 years of a person's life can be just as expensive or even more expensive, then it's not such a bad price to pay, considering the amount of time you would have saved from having to go do blood transfusions every month. People unfortunate enough to be born with thalassemia will get to live comfortable lives just like the rest of us. And so, even when it doesn't seem like it, the science and advancements in our world are growing at a startling rate. Soon, no longer will patients' lives have to be anchored near hospital premises, and nor will the hallways of vital medical centers be packed. A physician's role will undeniably be transformed through knowledge of genetics, moving away from prescribed medications to viral vectors. The treatments administered in hospitals will be increasingly quicker and, most importantly, permanent. It gives humans a chance to be rejuvenated and be reborn with new identity before any pain or suffering starts to settle. Here on Earth, we're giving humans the greatest gift known to mankind: time. And so the question really is, what are you going to do with your splendid gift?